And thank you everyone online for attending as well. It's my pleasure to introduce Christian Madsen, who's visiting the BSU and visiting my group for a couple of weeks. Christian is joining us from Melbourne, where he's a PhD student, final year PhD student in the theoretical system biology group, which I have some very strong connections with when it used to be in the UK and is now at the University of Melbourne. And Christian's going to be talking to us today about the topological properties of the protein universe, which is a very grand title, which I'm sure he'll explain to us. I'll hand over to you, Christian.
This title, Michael came up with it, so I don't know if that makes sense to you.
Yes.
So I have a background in biotechnology, mathematics, biology, and now computational biology. So I want to give that context. And final year PhD student. So I've been doing a lot of work with topological data analysis recently. And here's a big project on proteins, the structure of proteins that I have been working on with these lovely people. Especially Agnese has been a mentor of mine. I'll remind you first how for proteins, the shape is quite important. I'll say it dictates their function. So we thought it would be interesting to look at the shape with some topological data analysis. And for that, we have so for proteins, there's a lot of sequences out there. There's a lot of sequencing data for structures. It's a little bit more sparse. There's about 220,000 structures in RCSB, which are generally sort of these chains where we can see there's a larger protein and then we have a subdivision of it. So after AlphaFold came out, we thought it would be interesting to work on that. So for the uninitiated, AlphaFold 2 is a prediction of protein structures that have far outperformed what has been done previously. They're still not perfect, and there is a lot of predictions of their structures that are flawed in one sense or the other. Thankfully, there is often the ability to take good use out of their own scoring of their own quality. So to get started, we want to look at the data. We simplify to look at a point cloud of the backbone of the protein. So these are the carbon alphas of this protein. And then we do our topological data analysis. So within topology, there's something called persistent homology. And that's what we've been working with. And roughly in my words, I would say that it has to do with finding the shapes within data, in this case, sort of Euclidean space data, and looking at those shapes where they persist over different scales of the data. And each of these shapes will be sort of abstract, and there will be multiple different concrete ways of looking at these shapes. And that's where the homology comes in, that these concrete realizations are homologous to one another. So first, the data is a point cloud. Here, showing an example in just flat two-dimensional space. The first thing we do is, as I alluded to, look at it at different scales. And there's different ways you can think about this. I wrote approximate the data at different scales. You can think about it as disk or spheres sort of expanding in size over time, or you can think about it as, well, these are points, so they don't really have a size. So we're looking at all the different sizes you can imagine them having, or however you'd like to think of it. But then from this, we create a nested sequence of simplicious complexes. So for this, we have the edges and the sort of triangles here in the T2. So these are simplices, right? There's different dimensionalities of these. So a zero-dimensional one would be point, and then you have one-dimensional for a line, and then a triangle, and so on. So it is a generalization of the concept of a triangle, you could say. And if you take a number of these together, then you have a complex of simplices. And I refer to it as nested because, as you can see, as time progresses, what has been before is always sort of contained in the next time step. So, but yeah, as you can see, as these disks overlap, then we're connecting the dots and so on, until we get to a point where everything is trivial and everything's fully connected. So how to do this? There is a number of ways you can do this sort of thing. It's called a filtration, and my understanding is that the most sort of accepted as being correct in a way is the check filtration. So that's the first one shown over here. So you can note that there's 3 disks that are connected by lines, but because they're not all overlapping, we don't connect a face here. And you can also note that there's 4 points all connecting each other, so you get sort of a tetrahedron here that's flattened. But for computational reasons, this is not that commonly used. It's a bit more expensive than the Vittor's RIPS, which is the next one that people are generally using. So most of the software out there is for the VR, where they have made it more feasible to compute by simply saying that if you have dots connected, then we just connect the whole thing, not to worry about computing the overlap of all of these things. But then there's also a third option here that we found to be quite interesting. And I will spoil that this is the one we ended up using, which is the alpha complexes, where it has its own sort of shortcomings in that it has a limitation to how many large simplex you can create, right? So in this case, you see that we haven't created a 3 simplex, we only have a 2 simplex, and yeah, for this 2-dimensional space. And it also does compute it doesn't close this area above here like the VR, so it has some benefit in accuracy, you could say, if you consider the check more accurate. Yeah. So moving on, from such a analysis, the general output you get is a set of what's called homology classes. So these homology classes, in more regular words, are counting the holes in the data. And I think I should use the pointer here. So there are, you have a concept of a hole here, and you have another one over here that exists here again. And these are the same hole, which, so there are essentially 2 different homology classes, and then you have a concept of birth and death when they appear and when they disappear. And you can visualize that in just a diagram down here, which is a common way that people sort of end their persistent homology analysis. Sometimes you take it a step further, where we decide on a subset of the data to represent these homology classes, and we call this subset cycles or cycle representatives. We often call them just representatives for short. And there's a reason why these aren't always included, partly that they're expensive to compute, but also because there's not one way to select them, as you might be able to see here, where there's these 3 examples that I'm showing, but which one is the correct one to illustrate this hole up here? Is it the innermost, the outermost, and also even if you choose innermost, then there's different shapes at different time points throughout its existence. So yeah, for this reason, we sort of have gone with an approach of we will go with what is most computationally feasible because of the scale of the project, and then validate with the biology that this is still meaningful what we're getting out of it. So this leads me to post-analysis, where we came up with some further things in a pH pipeline that we devised to create some outputs that we felt were more interpretable to people. So these representatives, as mentioned, can be a bit noisy, and they're representing something a bit abstract. So we then describe them as a hypergraph, which is simply just a number of sets. So completely the same thing, but it allows us to think about it with regular graph data analysis, where we then can do centrality calculations, and we can do clustering. So for the centrality calculations, that gives us then a scoring for each of the points, so each of the amino acids, where we can say this is how important these points are. And yeah, so topologically important points is something we get out of this, where it's since these hyperedges are weighed by the persistence of each of the each of the representatives or each of the homology classes, we get out something that not only weighs or describes the importance of an amino acid in terms of is it a member of a lot of sort of features, topological features, or is it a member of large ones, but also is it a member of ones that persist for a long time? So it's sort of encompassing all of these things. And for the clustering, it also helps us with a number of things. Like if you have a lot of noisy small ones, they don't become necessarily something we have to look at. A lot of noisy representatives. And also because there'll be a lot of overlap between these representatives, and the clustering only assigns a vertex to one specific cluster, so we get a nicer sort of subdivision of the protein. So a note about computation. One of the main things about this project was that it was interesting computationally in terms of the scale of the project. So we had, I did a number of benchmarkings here to the very left in terms of time and memory usage of all the main tools I could get my hands on for calculating something like this. And first off, not all of them compute the representatives, so it's not necessarily something we can get out of it, which we require. And we also found that what we were working with for a long time initially, even given the large amount of computational power had available, it would have taken 2.5 years with an estimate. So it also wouldn't be feasible if we just did it the initial way. So the benchmarking definitely helped. And then we decided on the Ripster JL, which is a great package in Julia. And we went for the alpha filtrations that turned out to be significantly faster than other options. The estimate for that was still 1 month and 18 days. And I did get it further down to less than 3 days due to some other performance tweaks. And so I wanted to give some credit to that package and their work. They essentially found a very fast way of computing these representative cycles by first computing something else called representative co-cycles, which are cheaper to compute. But as you can see here from the image, they're less intuitive of how they're representing the data. Okay, so now to the biological results. I'll start with some of these voids, as we like to call them. So these are the two-dimensional cycles. So all the examples with the illustration from before were one-dimensional, but you can imagine how this sort of still can be extrapolated into other dimensions. And we did this work for 1- and 2-dimensional. So here we thought about the voids aren't exactly literal voids or gaps in the protein, but there's something like that. So we thought it might make sense that if you think about something like temperature, then potentially you can consider temperature something that sort of adds, if you have an environment with a high temperature, there might be some sort of added pressure around the protein that might compress these gaps a bit. So we looked at thermophiles versus the equivalent protein in mesophiles. And we did find exactly what we were hoping for, which is that there is a tendency for the to have fewer large voids in the thermophiles. So there's A, illustrating one example of this, and B is all the numbers for all the enzymatic classes that we looked at for this. And then C was a sanity check, because we thought, well, since we're only looking at the backbone, what if this is really explained by something else? Like, what if it's the residues that are different? So if you have large residues, small residues, that would make a huge difference for how filled out these voids are. So we wanted to consider which amino acids are present in the voids. And we did this with the void centrality. So this measures not only importance of the amino acids, but also, well, how much are they present in these voids? And as you can see, there's no difference between the amino acids in meso and thermo. So that was good.
Sorry, Christian, can you say a bit more about B? Because I'm not sure I understand what.
Yeah, so B is a bit of a creative way of drawing a figure, but we thought it looked pretty. So across the length here, you have 100% of the proteins, right? And then we have divided them into 4 categories based off of the size of the voids. And we are showing that sort of with the different molecules that are approximately that size to give you a sense of scale of what would fit inside these voids if they were perfect voids. And then you see the how big of a fraction of the whole that it belongs to each of these categories of sizes of voids. And then we added some, the gray is the standard errors to indicate how significant this is. Yeah. And then moving on, there was a result here where we did the clustering that I talked about before on these same voids, and we find that the boundaries between these clusters are enriched for binding sites, as you can see here. And my intuition for that is that if you sort of imagine a protein that has two main globular structures, you would get a lot of void clusters within each of these parts separately, and the point between them could be sort of hinge point. So it makes kind of intuitive sense that the interesting binding dynamics would happen in that point between, but it's not like these. I think I also want to remind you, the idea isn't to make necessarily a binding predictor with this work or some other predictor of something. It is a general analysis of what you can see that is intrinsic to the shape of the protein. So if you want to do a binding predictor, you might incorporate this, but it wouldn't be the whole story. Yeah, so moving on to the topological clusters for one-dimensional cycles that we also looked at. So for this, we compared them to a Cath database. So the Cath database, my understanding is it roughly divides a protein into different domains that are supposed to sort of have independent lives, like the independent folding mechanisms or something like that. And yes, so to the left, we have the Cath domain of a single protein, and to the right, we have our topological cluster. And this was sort of the picture we were seeing again, where there is a sense of they match pretty well, and there's a subdivision happening in our topological clusters. Yeah. And B is then showing you the general picture of all the data with a homogeneity score of how well these two divisions of a protein fit together. Yeah, so I thought it might relate to folding, and then it would be interesting to look at some actual folding data. Unfortunately, there's a lot of simulation data for folding, but not a lot for actual experimental resolved information at different time points during folding, because it's a very fast process. But I did find one example of this with horse epomyoglobin. So we had two different time points here where the color is indicating, again, the topological clusters. These semi-transparent regions are yet folded. They are. And we were happy to find that it fit really well with our clusters. The first thing that folds is essentially this large blue cluster. Then, not as clearly, but at the center image, it's the alpha helix at the bottom that gets folded, which has both blue and red parts, so it sort of follows naturally. And then we don't know exactly what happens in between these steps, but then the rest folds. So it's just one example, so it's not a strong result at all, but it was nice to sort of see that it fit in the one example we could find. So moving on to centrality scores. And for those, we felt that it would make sense that if we're talking about importance, topologically speaking, of amino acids, then how would this relate to biology and real life? And the idea was that, well, it would probably relate to if you mutate this amino acid, then that should have a big impact on the structure of the protein if it's scored highly. And this was also what we saw. So we found some data for damaging and neutral mutations, and there was the correlation you can see here with our scoring of importance. And then also as a sanity check, I know a lot of the feedback we've gotten is criticism of the trustworthiness of the predicted structures. So here's also a comparison to the equivalent structures that are solved from RCSB. So we looked at the centrality score as an example of something we could do a correlation on between the experimental and the predicted structures, and they fit pretty well. But there's also a note here, which is that you'd probably imagine that the AlphaFold would predict well on the structures that are already solved. Some of these structures are from after AlphaFold was made, so there's, you could say it might not. So that's a better test of its quality. But there's also a note to be made about the solved structures aren't also the perfect ground truth. You could have a protein that would have parts that are moving and so on, that might be different conformations or slightly different angles of the protein that could both be true. And moving on to the final results, we wanted to do something that essentially used all of the data, since we've done such a large sort of analysis of a lot of data. So we took the 214 billion proteins, we filtered them by quality. So it's only the high quality structures used. So that's 167 million, given the AlphaFold's own sort of scoring of quality. And then we did this analysis here, where because it's so much data and it's such a high level, the way that I felt this made sense to do was to do it at the comparing domains of life. Yeah, so the first one, subfigure A, is it says representatives per residue. So the representatives are either the loops or the voids. Right? So you can think of this as, if you have a given residue, a given amino acid, how many of these loops or voids is it a member of? And you can see the distribution here for all of the proteins for this. And this is also important to note that these figures are normalized so that each species is weighed equally. Otherwise, you'd get a very different result. If you did it for unweighted like that, then you'd get it for the model organisms would take the bulk. You had a question?
Yeah. So so on the x-axis of the representatives per residue, you have like integer numbers.
Yeah, it's not integer numbers. These are floating point numbers, but the scaling is just showing you where 1 and 2 and so on. But this is 2.0, right?
This is 2.0.
I'm saying 2 is 2.0. It's not integers. This is a fraction, right?
I was just having trouble understanding what, like, being part of half of a void. Is that what it means? So
right. Well, it's a division of the size of the protein, right? So I'm saying per residue.
50% of the protein is in the loop or something.
Exactly.
Gotcha.
Yeah.
Okay.
Yeah. So it's per protein, right? So per protein, and then normalized by the species. Okay. And then the second figure here is the maximum simplices per protein. So for loops, this is the most intuitive one. For this, it roughly corresponds to saying, take a given protein, take the largest loop, what fraction of the protein is within that, is captured by that, right? So 50%, and you can see it sort of goes to 1, so 100%. So if you had some loop that captured all the protein, you'd have a value out there, right? So this one is more intuitive than the voids one, because the voids, you have 3 in each edge, hyper edge. So it becomes a little bit more tricky, but with a scaling factor, it would be roughly equivalent to that idea. But the interesting here to me is that we're seeing a stark contrast for the eukaryotes. They're highly different, and all of these 4 figures are pointing to the same story, that their proteins are topologically more complicated, which I find quite interesting. It also kind of makes sense, I think. Yeah. So then there's a bit of a open question of, well, why do they seem to be more complicated like this? Is it something with post-translational modification that is more common with eukaryotes, or something with intrinsic noise, which I also found some, I have the impression that it is also always more common with eukaryotes. So I haven't looked into that yet, but yeah. And then I want to finish with an idea that not done yet, but something I thought about, which was that often proteins and through them species are compared on sequence, right? When you're reconstructing trees, or if you're comparing proteins in general, you do this, and it makes sense because you have the data and so on. But it's the sequence isn't a perfect predictor of the function. So what about using something like the structure, but then you have the problems of availability and computational cost and so on. And you also have some issues of, well, if you just take two structures and you superimpose them, well, that's not invariant to bending, right? If you have some section that is completely bent, then you're only superimposing half of the structure, so this is not perfect either. But this is where the idea is that the topological comparison is invariant to some degree to that, as long as it doesn't create new features. So this would be a better tool for that. And as long as you then accept that you have the structures available, so that requires that you accept AlphaFold as good enough, then you could do something equivalent. You could do some tree reconstruction or similar using the topology. So then the question is, how would you compare the topologies? And for this, there are a number of ways. There's the very established Wasserstein distance that compares the persistence diagrams. But it's also costly. Then I have some colleagues, Stephen and Nisa, that are working on using optimal transport for topological data analysis. They call it topological optimal transport, where optimal transport essentially finds the optimal way of moving one probability distribution to another one. But this is also very costly. It's very cool for small examples, but for this scale, it very well. But then I thought about, what if we use some of our very simple outputs of our pipeline, something like the centrality scores, which is just a bunch of numbers along the sequence, right? So we're already doing alignment on sequences using their letters. Why not do something similar just on the numbers from the centrality? So then I looked at two proteins that are very similar to get the idea if this would make sense at all, trying to see if their centrality curve along the sequence would be comparable. And if you align these sequences, it matched up in this case. So I thought maybe this could be a useful way. Of course, this doesn't mean much if it's just that these mutations that differ are small things that doesn't actually change the shape that much. So this requires more work to see if this is useful, but that was an idea. Yeah, I think
that's really interesting. So because you're kind of thinking you'd have like a topological blast in the end or something.
Exactly.
So you're trying to determine whether or not two proteins are homologous on the basis of their topological properties rather than just the sequence.
Yeah.
I mean, I guess one question then is, so you do that, and I mean, you come up with some answer, you come up with some ranking of relationness.
Exactly.
How do you then check? How do you then check that?
If you have the rankings and you have the best match, right, then you've done the large analysis that is where you have to do this trick to make it feasible computationally, but then you can do more, costly comparisons, right? Then you can do like the Wasserstein, the optimal transport, or something like that to see if they correspond really well, or they're looking at them or superimposing them or whatever, right? And this would be very good for something like convergent evolution. That's something I was excited for it to be used on, where you have the same function that evolved from two different places. The sequences are highly different, but they might look the same, and maybe there would be connections here for that. But yeah, I mean that's all the slide. So thanks for listening.
Before we take questions, Pantelis, can I ask a favor of you? I don't know how to work the questions in the web.
I have laptop here, so if there are any questions online, I should be able to unmute. So if people raise their hands or type, I will, yeah.
So I will start with, so I mean, so it's really impressive, right? Because you've taken this huge collection of proteins, and then you have applied this TDA pipeline to them, and you've come up with this massive kind of characterization of all the proteins, right? This is, and you've managed to do that in 3 days or something, which seems incredible. And then on the basis, sorry, I'm just kind of summarizing my answer.
Yeah, no, it's great.
And on the basis of all of that, you're looking at different kind of classes of proteins. You're looking at various ways in which these statistics. So I think of TDA as basically just giving you a collection of statistics You're looking to see if these statistics are associated with various things, and so one question I have in general about TDA, and I think it's relevant here. So when you're looking at the comparison, so you're doing this void comparison between your two classes of proteins, and then you were saying that. So what was it? You had your was it your thermophiles and your mesophiles, and you kind of so you come up with this, and you look at the sort of distribution of the statistics in one, and then the distribution of statistics in the other, and then you kind of say they are different, right?
Yeah.
But in order to do that kind of, this is the stats unit.
Yeah.
What is the test? What test are you doing? So is there a formal statistical test that you're doing, or are you more kind of saying, well, that the distributions look a bit different, or are you formally testing this? And if you're formally testing it, then how are you doing that?
Yeah. All I'm doing is calculating the standard deviations of these points of difference here. I mean, that's something that I could get out of being here, is to get more formal statistics tests. But I think a lot of these cases, it is clear enough that you don't need to test it. It's not on the point where you're unsure if it's different. And I think also the idea isn't necessarily that I have to prove that they're different. It's that it should make sense what we're getting out. It's more the validation that people will understand that this is useful and it's meaningful, right?
Well, I guess the concern would be that if you sort of randomly take half of your thermophiles and split them into two sets and then do an analysis of those, then by chance there will be some difference in the distribution of the statistics that you get from those sets. And I guess it'd be nice to know for sure that what you to be able to say whether or not what you're getting is
I talked to the others about doing bootstrapping to just shuffle it around and to see if we would get something out at random. I think we had a decent amount of proteins here, so I think it works out fine. But part of it was also that when we were looking at the 214 million or the 167 million, the idea wasn't that at that point you don't need statistics anymore because you're not describing a sample. You're describing a sample, but it's a huge sample, right?
Yeah. So you are so your pipeline is in my calculating statistics.
Yeah.
So summarizing this huge data bank of proteins into a collection of numbers, basically characterizing each protein. But then, and I agree, at that point you've just calculated statistics, right? But then as soon as you want to do any kind of comparisons, that's where I feel you need to be doing some kind of statistical modeling.
Yeah, well, that's great. Then we can talk about doing that.
Right. I have more questions. But,
Thank you very much for the talk. Could you please explain, like in one of your first slides, you were explaining the different graph structures. So you had 3 of them, A, B, and C.
Yeah, this?
Yeah, and I wasn't sure I got the difference between the second and the third one. Could you give me?
Yeah, so the second one was the Vittori's rips, right? So it is what you have at this point. So you have some kind of spatial parameter or time, you can think of it, where the disks are a certain size for each of these points, right? So this is sort of fixed for all these scenarios. And what is then different is how you choose to connect things, how you choose to say that now they're overlapping, right? And in all these cases, the lines are pretty trivial. They're overlapping if the disks overlap. But then you have to decide how to do it if you have more than just 2 points connected, right? And you can do this as a number of ways. You can say that if you have these 4 dots connected, then all of them are connected in one big simplest, which is done in the 2 first ones. Or you can keep it more simple and you're computing something called a Voronoi diagram in the alpha complex, where you're sort of drawing lines in between these. I haven't had time to add it, but there's sort of like, imagine lines that is subdividing between these. And this makes it computationally like very easy to then compute when there are 2 simplices in the 2-dimensional case, but you can't go further essentially, right? It's just, this is also what's shown in the middle here. This is the alpha complex. So then there's like pros and cons, right? You're essentially artificially, if these 4 points here are equally distant from each other, it's artificial that we're drawing a line here and not the other way, right? So there's some cons to it. But partly this is also why we are doing the clustering, is that we're hoping that it sort of groups these together as one cluster, because even though that we're saying that these three are connected and these three are connected because they have two shared in each of the simplices, then we say the whole thing is connected. Yeah, does that explain?
Sorry for my ignorance about all these things, but my understanding is that the AlphaFold will give you representations in 3D.
Yes.
What you're doing here, is it 2D?
I was just trying to draw an example in 2D because it's easy to, yeah, I can't draw ticks in 3D very well. So this is for illustrative purposes, but everything is done in 3 dimensions. That's also why, yeah, so this is 1-dimensional. And that's why I wanted to make the point how these voids are the same concept, but in an extra dimension. Right? Yeah, everything's 3D clear in space.
I have 2 more questions, I think. So one is about the clustering that you were just mentioning. So this is my ignorance. I don't really understand how clustering works in this context, but in particular, how do you decide the number of clusters?
Yeah, so that's the thing. We were thinking about how to cluster this for some time. There's a lot of work out there to do hypergraph clustering. A lot of it, most of it does not, you can't compute it in a reasonable amount of time. It's crazy. Sometimes they will even, some software will create a regular graph and hypergraph that has the number of dimension as big as the largest hyper edge. And then you have like null dimensions or something like that. It gets very crazy when you try to do graph stuff on hypergraph sometimes. And often you have to decide a number of clusters beforehand. And we didn't really want to do that. But we found something where you don't have to do it beforehand. It sort of optimizes a concept of modularity. Which, yeah, hard to define, but that's also why we did all the biological validation testing afterwards. It wasn't just to find like, you know, saying this thing about topology and biology. It was also to validate that our approach was sound, essentially, that this choice of clustering was meaningful by seeing that biology was meaningful. So it goes kind of both ways.
Vaguely, does modularity mean that within cluster similarity is high and between cluster similarity is also?
Yes. So I try to illustrate it here where it's in a simple case, it would be identical to the hyper edges. And then as you make it more complicated than this, then you can imagine how if you have 2 hyper edges that are highly overlapping, then that's one cluster, right? So the whole thing just gets combined. If you have 2 that are completely separate, then they're separate, right? And if you have 2 that are a little bit overlapping, like these 2, but not quite, right? They're mostly separate, then they get cut, right? That's sort of the idea.
And my second question was going to be, so suppose I want a piece of your results. I don't want to have to rerun all of your pipeline again.
Yes.
Has this been deposited? Has all of the TDA output been deposited somewhere?
Yes. So we have a number of ways to access this. I've done a lot of this summary stuff that is visualized here, for instance. A lot of these values have been computed, and there's stuff per species and per protein and so on, and per domain and per all the different taxa in between that have been pre-computed in a Postgres database that you can download. It's only like 13 gigabytes something. So you can download that if you have the right, the 15.2 Postgres, then you can install that. Then you can very quickly, with a small computer, look at the summary statistics. And then if you want to access the protein structures, we have a large download where you can download, we've divided into different ways to manage the data, the scale of it, because it is very large. So we have HDF5 files that we have put a lot of this into. We also have it as JSONs. You don't know what it is? It's essentially a small file system as a file, which means that you have partial I/O you can read right to it without having any cost. So even if you have a certain number of files, you can just read one of them. It doesn't cost you extra that there's more before and after. Yeah, so it's very useful for things like going in and if you just want to read if you just want to read the amino acid sequence of one protein that we have included in the file, right? But you don't want to read the whole file out. You can do that without reading anything else. So it's like if you want to do analysis of all of the proteins, all of them, for a specific thing you care about, you don't care about the rest. You only read that part of every file, and it doesn't cost you anything extra. So it's quite a good storage, but the whole thing takes up a lot of space. You can also download certain. So it's also divided by the taxon numbers with like folders for the 3 digits and. But we also have plans to put this. I haven't had time to do it yet, but we have plans to put this as a Postgres database on a server where you can then query and then search for subsets. Yeah.
And then one day you'll have a server providing you with your topological blast as well, so that people can do that. Fantastic.
Yeah, so that would be cool.
Very interesting work. I'm wondering, would you provide some metric, say, now AlphaFold 3 just come out this week, say, okay, this one you don't do very well, or something like that. And because from an applied perspective, another thing we are interested because within each protein, you got many sections of them are peptides. We know it's like when you do the sequence analysis, they should match that. But if you have a larger pool, many of them just say your protein is from not a clear cut, this is this one. More or less, you have a mass spectrometry, say, okay, presumably this segment belongs to that one. What chance you make a mistake in those scenarios?
Mistake about, sorry, what?
Yeah, so 2 questions. One is like whether you have a kind of objective metric, like informing your confidence over AlphaFold. And the other aspect is, say, you only got segments of proteins. Would you be able to align correct which one with confidence, which protein, say?
For the first question, how much we have confidence in AlphaFold 2?
Yeah, if you're measuring against that, you have I believe you have some kind of.
Yeah, we have a metric. I mean, they have a metric. They have an average PL/DDT scores, they call it. I think it is, if you look at the proteins that they couldn't predict, it's very obvious. They look just like part of it looks like a protein, and then the rest is just like a long arm that just goes out. Thankfully, it looks kind of terrible if they just sort of gave up if they can't predict it. But I mean, it's not the ground truth, of course. But yeah, I mean, I think it's pretty good when they have a template and so on. The second question was
more like you have a universe of segments of proteins. Would you I don't know, you need to predefine your protein sequence to infer the structure, or you can't really work from your data in more.
If we can infer the structure from segments. I mean, we haven't done anything like that, but that would be cool.
Now we got the data from various platforms who do peptide association.
Yeah.
We have a strong belief they belong to that protein. We believe they are not, because the signals we saw it just doesn't align so well. So it's very practical. Obviously, if we define, okay, this is the sequence, and we're going to read this the whole alphabet. We think this 3D like that. It's very much you predefined your scope already.
So is it right that everything you're doing is at full protein level?
Yeah. I mean, full protein, we're doing it on the chains, right? So what they have predicted is chains. So it's subunits of the proteins. Yeah, we haven't done it on peptides or something like that.
Okay.
And often at that scale, you wouldn't have interesting topology. It might be too simple.
In your last slide, you mentioned, well, maybe it was not the last. Yeah, you said like comparing topological characteristics, and you said that it's not possible with these methods with the current scale. Now, I'm just wondering what the scale entails here. Like,
Are you talking about? I think it was around this slide here.
yeah,
I was talking about the scale in the sense of if you want to reconstruct a tree of life for all species on the planet. That's essentially what I was trying to do. I haven't included here, but I have a figure where we're trying to sort of illustrate the topological complexity and richness across all species, right? And in order to do that, that would be too expensive to do with Westerstein or topological optimal transport. But that's something you might be able to do with comparing the centralities. So then you'd be able to, do like a topological angle to how you think the sort of tree of life is, right?
the centrality would be like a summary.
Yeah, it's something that is cheap enough to compute on. But then, so that would be maybe useful to give a hint at how the tree of life is according to topology. That's the idea, at least, if it works.
Were there any questions online?
I cannot see any questions online.
In which case, I would just end by saying I think the title is justified, because given the scope of what you're doing, I think yeah, Michael is right to have a bold title. Let's thank Christian again.